So our next speaker is Alberto Anel. Alberto Anel is a bachelor in chemistry and PhD in biochemistry by the University of Zaragoza. He started its interest in immunology in its postdocs, first at the Medical Biology Institute in La Jolla, California, and at the Centre d'Immunologie de Marsella. Back to Spain, he is now director of the Biochemistry and Molecular Biology Department of the University of Zaragoza, and principal investigator of the research group Immunity, Cancer and Stem Cells, where leads the studies of effector mechanisms of the immune system by cytotoxic T lymphocytes and natural killer cells for cancer immunotherapies. Alberto.
Thank you very much for the introduction. So this, how this works. Okay. And the marker. Where is the marker? Okay. So I'm very happy to be here, and I also would like to thank Maite Galvez for inviting me to this forum. And I'm trying to summarize in a few minutes the lines of research of our group that are more related with the interest of this forum. First of all, our group is interested in the identification of immunogenic chemotherapies, especially for the treatment of multiple myeloma. Multiple myeloma is a hematological malignancy, normally with bad prognosis, that until 2004 didn't have a good treatment, an efficient treatment. In this year, it appeared the first proteasome inhibitors, that it was bortezomib, also known as Velcade, that gave new hope to these patients. Now we are studying the cell death mechanism induced by a second generation proteasome inhibitor, carfilzomib. And what we want is to combine carfilzomib with other drugs. First of all, to kill better the multiple myeloma cells, but also to induce immunogenic cell death. That will, if we arrive to do so, then we could combine these chemotherapies with immunotherapies, like, for example, anti-PD-1 blocking antibodies that are working very well in other types of cancer, but not so much in multiple myeloma. The drugs with which we are combining carfilzomib are chloroquine, DBQ, and CB5083, both of them BCP inhibitors that would act as ER stressors. Then we measure danger signal generation in dying multiple myeloma cells, especially calreticulin exposure. And then these danger signals are hoped to induce dendritic cell maturation and phagocytic activity that would generate finally an efficient anti-tumoral response. One of the most important aspects of the research of our group is the collaboration with the hematology groups in the two hospitals in the town that allow us to have samples from multiple myeloma patients, normally medullary aspirates. On these samples, we determine several markers to identify the multiple myeloma cells, and also we determine the basal level of calreticulin exposure. We also analyze the tumor microenvironment, especially the presence of effector anti-tumoral cells, such as NK cells and CD8 T cells, but also of other immunosuppressive cells, such as CD4 T regulatory cells. We have also observed that two types of patients exist. Ones that have a low calreticulin basal level, and others that have a high calreticulin basal level. And we have demonstrated that these patients that have a high calreticulin basal level has a tumor microenvironment more immunologically active, with a higher percentage of effector NK cells, and also of CD8 T cells, and with a lower percentage of immunosuppressive Tregs. We also want to validate the previous in vitro studies that I described in an in vivo multiple myeloma model, in which by injecting this multiple myeloma murine cell line in immune-competent mice, they develop a real multiple myeloma illness. We will test then the best immunogenic combination of the drugs that we have tested in vitro, and then we will combine then or not with anti-PD-1 antibodies. We will follow disease monitoring by determining the multiple myeloma protein and also multiple myeloma infiltration in bone marrow and spleen, in vivo imaging, and survival. And if this in vivo model works well, then we will also analyze, we'll do a similar work in the humanized NSG mice, in which human multiple myeloma cells will be injected. Another way of killing tumor cells would be to take advantage of their special glucose metabolism. As you know, many tumor cells rather perform glucose fermentation instead of using mitochondrial oxidative phosphorylation or OXPHOS to obtain energy. In a previous work, we have characterized this cell line that we call L929DT, that derived from an original transformed fibroblast model, L929, that spontaneously detached from the culture dish and from the rest of the cells. And we thought that this could be a good in vitro model of metastasis generation. This cell also lost MHC class I expression, making them refractory to CD8 T cell elimination, and also had signs of an acute fermentative phenotype. Now in the project that we are developing now, and in collaboration with Raquel Moreno from the GenoxFOS group, we have determined the mutations that in the mitochondrial DNA that suffer these cells, if we compare with these ones. There are mutations in the ND2 component of complex 1. We have observed that this causes a defective supercomplex assembly in the mitochondria. This would be associated with an increase in ROS generation, but these cells increase very much the ROS detoxifying activities, catalase and superoxide dismutase, making them resistant to oxidative stress. These observations would fit in a recent model in which it is proposed that cells that detach from a primary tumor mass, normally they suffer oxidative stress and die. But the cells that finally arrive to make a metastasis do so because they develop mechanisms to resist this oxidative stress, and then they are able to travel and to form distant tumor niches. Okay, so with that, with this data, we would just describe a new mutations in the mitochondrial DNA that would explain the generation of metastatic cells. So perhaps we would know better the cells that are going to kill us when we had a cancer. If we had not also described that these cells, but not these ones, are sensitive to cell death induced by a simple metabolic drug, dichloroacetate, that forces cells to use oxidative phosphorylation to obtain energy. And in addition, we have observed that DCA increase MHC class I expression in these cells, so suggesting that DCA-treated metastases would be more sensitive to immunotherapy mediated by cytotoxic T cells, such as the anti-PD-1 antibodies that we mentioned before. Another immunotherapy that has a lot of future is the use of adoptive transfer of natural killer cells, of NK cells. And our group, and that of Julian Pardo here, and the group of Martín Villalba in Montpellier, we have developed some protocols to activate and expand these human NK cells using EBV-positive lymphoblastoid cell lines like this one. And we have now tested two combinations of cytokines together with the activating cells, IL-2 and IL-15 in the absence or in the presence of interferon alpha. In both cases, we have arrived to NK cell expansions that would be enough to treat one hematological patient with cells from one healthy donor. We have tested these expanded NK cells on cells from 28B chronic lymphocytic leukemia patients, and we have observed that most of the patients, all except these 4 cases here, are sensitive to these expanded NK cells, showing the feasibility of using expanded NK cells for the treatment of BCLL. And now we are making a similar work, but using samples from multiple myeloma patients, as I have described before. And finally, our group is interested from some years ago on the mechanism of tumoral cell death induced by granulysin. Granulysin is a protein that is expressed in activated cytotoxic T lymphocytes and also in NK cells. And then we have described before in our previous publications the mechanism of cell death that induces in tumoral cells, that involves interaction of the protein with the plasma membrane, loss of ionic equilibrium, increase in calcium, intracellular calcium concentration, and so activation of the mitochondrial apoptotic pathway that finally leads to tumoral cell death. This is the structure of the protein. And more recently, we have demonstrated that recombinant granulysin is efficient in the treatment of 2 models of in vivo development of human tumors in anemic immunosuppressed mice. The mammary adenocarcinoma MDA-MB231, in which granulysin stops tumoral growth, and the multiple myeloma NCI-H929, in which granulysins completely eradicate the tumors. And now we are optimizing, in collaboration with Ramon Hurtado, the production of this recombinant protein in the yeast Pichia pastoris to avoid the presence of endotoxin in the final preparation, like LPS that was present in our previous preparations in Escherichia coli. And these are our financial support. And this is the picture of the group like it is right now with the 2 other PIs, Isabel Marzo and Javier Naval. Thank you very much for your attention.